Bismillahirrahmanirrahim. Angiogenesis, that is the birth of new blood vessels, is discussed in this video. Following are the references for this video. Angiogenesis refers to the development of new blood vessels from existing vasculature. This process is vital for tissue repair and wound healing at the injury site. Collateral circulation in ischemia, for example, heart or peripheral tissue, and also important for tumor growth beyond the original blood supply. The pathological new vascularization also occur in some diseases such as diabetic retinopathy. This angiogenesis is a stepwise process. First, there is vasodilation and increased permeability that is triggered by nitric oxide and vascular endothelial growth factor or VEGF. It prepares vessel for new sprouts formation. Then the parasites are detached and there is lysis of the basement membrane or breakdown of the basement membrane. The parasite detach from the abluminal surface of the existing vessels, and extracellular matrix breakdown allows for endothelial sprouting. There is endothelial cell migration occurs, and this is uh, this process occurs as endothelial cells they move toward injury signals guided by growth factors and extracellular matrix cues. Then next step is proliferation of the endothelial cells. They cell just behind the tip of the migrating front, they multiply to create a cellular scaffold. Then there is capillary tube formation, and the remodeled endothelium forms luminized tube for blood vessel. Then there is recruitment of peri-endothelial support cells. So the parasites for small vessels, they are supporting cells. Smooth muscle cells are supporting cell for the larger vessels, and then there is connective tissue deposition that stabilizes the vessels. Now the supportive tissues are there and the sprout has formed. So there should be suppression of further growth and basement membrane deposition. So the further growth is suppressed and basement membrane is deposited. There is proliferation of the endothelial cells and migration that now stops once the mature vessel is formed. The basement membrane is re-established for the stability of these endothelial cells, and hence the stepwise process of this uh, angiogenesis occurs. Various growth factors are associated with this process, and each or there is overlapping of one growth factor for the angiogenesis, various steps of the angiogenesis. One such factor is vascular endothelial growth factor that acts on various steps of this angiogenesis. It drives endothelial cell migration and proliferation. It promotes vasodilation via nitric oxide, and there is also vascular lumen formation. The fibroblast growth factor is responsible for stimulation of endothelial cell growth and fibroblast and macrophage migration, and it also enhances epithelial wound healing. Angiopoietin 1 and 2 are linked with the endothelial cell migration and proliferation of endothelial cells. They angiopoietin 1 binds to Tie2 receptor, that is a tyrosine kinase receptor, on the endothelial cells and promotes its maturation. The platelet-derived growth factor specifically acts in the step of recruitment of peri-endothelial supportive cells. It also recruits smooth muscle cells or pericytes. Then transforming growth factor beta, it suppresses the growth, further growth, once there is an interaction between the endothelial cells and the pericyte. It suppresses further growth and it suppresses migration also. It also enhances production of the extracellular matrix protein. The notch signaling pathway is linked in this stepwise process of the angiogenesis, and this signaling pathway interacts with VEGF to control branching and spacing of the new vessels. Then there are extracellular matrix protein and integrin. They act as a scaffold for the growth, and integrin on the endothelial cell, they mediate adhesion and signaling. Matrix metalloproteinases, they are MMPs, they degrade extracellular matrix to permit vessel extension and remodeling. So there is clinical relevance of understanding of these steps as the therapeutic angiogenesis occur. It stimulates new blood vessel to improve blood flows, for example, in ischemic heart disease. Then anti-angiogenic therapy, it is also used to block vessel growth to starve tumor or abnormal vessel growth that occurs in diabetic neuropathy. So it is a part of the treatment to stop the retinopathy that is occurring in the diabetes. So the angiogenesis or growth of the capillaries has got a significant role of the healing, as the granulation tissue has the highest density of the capillaries in any tissue, ensure the delivery of the oxygen and nutrient. And this angiogenesis is it occurs by the sprouting of endothelial cells from existing venules, and they form new capillaries. So this granulation tissue, granular appearance and its name, it is due to proliferating capillaries. Infrequently, vasculogenesis de novo occurs, and it is due to the effect of endothelial progenitor cells. Here, it is termed as vasculogenesis, and this vasculogenesis is more common in development. So the uh, hypoxia inducible factor alpha is one that is released, and it causes the release of VEGF, vascular endothelial growth factor, that is responsible for angiogenesis. And the transformation of the quiescent capillary endothelial cells to the activated endothelial cells is essential for the process to begin. And the important signaling pathway that is involved in this is a notch signaling pathway that that results in the vasculogenesis. So the initial event that is responsible for the vasculogenesis is loss or degradation of the basement membrane along with the detachment of the pericyte, and the cytokines and growth factors that are responsible over here are VEGF, fibroblast growth factor 2, transforming growth factor beta, and platelet drive growth factor that are released locally, and they also stimulate the growth. There is disruption of the basement membrane, and this is due to effect of plasminogen activator, matrix metalloproteinases, and integrin receptors. So all these three are essential for the proliferation and assembly of endothelial cells to form new vessels. So this activated endothelial cell, they also give rise to fibroblast growth factor, and they are also a source of VEGF, while the activated granulation tissue macrophages, they also produce VEGF. VEGF also comes from keratinocyte growth factor or fibroblast growth factor 7, that is derived from dermal cells, and from there it comes to the wound epidermal cells, and the wound epidermal cell then they produce this VEGF. Vasculogenesis occurs by various processes: chemotaxis, haptotaxis, and durotaxis. Chemotaxis is the migration along the soluble cytokine gradient, while the haptotaxis is the migration that is guided by extracellular matrix proteins. The durotaxis is a term that is used for migration that is derived by mechanical signals in the matrix. So there is migration of these cells, these endothelial cells. They migrate, and their migration may be stopped by transforming growth factor beta and PDGF, or they then proliferate, and VEGF is responsible for their migration and proliferation. Angiopoietin 1 and 2 along with fibroblast growth factor also play their role. They are immobilized over here. There is cell to cell interaction or contact forms, and basement membrane develops around the nascent vessels. The pericyte endothelial interaction and signaling from angiopoietin 1, transforming growth factor beta, and PDGF are essential to stabilize the vessel, reduce leakiness, and promote mature vessels. Immature vessels may be leaky, and there will be risk of hemorrhage and edema. So mature vessels are non-leaky, and if this extracellular matrix scaffold is not there, or growth factors are deficient, there will be presence of leaky vessels, hemorrhage, and ultimately apoptosis. So if even a single allele is lost in embryos, there will be lethal vascular defect that occur. So in vivo, this vasculogenesis or angiogenesis is initiated by hypoxia that triggers VEGF expression via hypoxia inducible factor alpha 1, that is the oxygen sensing transcription factor. MicroRNA expression also varies during healing, that is influenced by oxygenation, and We have already discussed the key growth factors that are responsible in angiogenesis. They are produced from the macrophage and endothelial cells, such as fibroblast growth factor and VEGF that occur in response to the injury. Epidermal cells produce VEGF in response to keratinocyte growth factor. Keratinocyte growth factor or fibroblast growth factor 7. So VEGF is critical for endothelial survival, differentiation, and migration. If there is a splice variant presence, it guides vessel branching via gradients, soluble or matrix bound. So FGF2, that is also termed as basic fibroblast growth factor, it promotes the endothelial cell proliferation, attracts fibroblasts and macrophages. Angiopoietin 1 and 2, angiopoietin 1 and 2, they interact with Tie2 receptors and stabilize the vessel. Transforming growth factor beta and PDGF, they suppress endothelial proliferation, promote extracellular matrix protection, while the PDGF recruits smooth muscle cell to support the vessel wall. The extracellular matrix and proteoglycans also play their role in angiogenesis. There is presence of heparan sulfate rich proteoglycans, for example syndicanes, and they are site for storage and release for VEGF and FGS. They modulate the receptor clustering and strength of the signaling and also influence the pattern formation of the capillaries. The extracellular matrix glycoprotein have got dual role. They are first a reservoir that store the VEGF and fibroblast growth factor within the basement membrane, and they also serve as a signaling scaffold. The cell surface proteoglycan regulate receptor activation and downstream signaling. This set of figure is collectively demonstrating the dynamic process of the tissue repair. From the macroscopic view of granulation tissue formation over here to the microscopic detail of angiogenesis and fibroblast activity to formation of mature tissue structure, scar tissue structure. All these are underpinned by cellular and molecular mechanism that are illustrated over here in the center one. So, in this gross picture, over here you can see that this is a skin wound that is covered with granulation tissue. This granulation tissue that is indicated by this arrow appear as moist red granular surface. The tissue fills the wound and provides a scaffold for further healing, including reepithelialization and remodeling. And over here in this microscopic picture, in this histologic picture, you can see that this granulation tissue is immunostained to highlight newly formed capillaries. The brown staining outline, this brown staining outline indicate the rich vascular network that is characteristic of granulation tissue. The endothelial cell proliferation is essential for supplying oxygen and nutrient for the healing process. Then a close-up histological or high power view of the granulation tissue It shows numerous proliferating fibroblasts. These elongated spindle-shaped cells are fibroblasts with are present that are present with newly formed thin blood vessels, and there are many or macrophages or inflammatory cells interspersed. This loose matrix forms the foundation of repair tissue. Then a later stage of the connective tissue formation is shown in this picture. Here, increased deposition of collagen fiber and reduced cellularity indicates the progression toward the scar formation. Blood vessels are more mature and less numerous. In the center, the gradual process of angiogenesis is depicted. There is first vasodilation in response to VEGF and nitric oxide. Then there is degradation of the basement membrane that allows endothelial migration, and later endothelial migration occurs, and this endothelial migration toward the angiogenic stimulus. Then proliferation of the endothelial cell occur, and this endothelial cells behind the leading front, they proliferate. Finally, later these endothelial cells, they form capillary tubes, and there is recruitment of pericytes and deposition of basement membrane occurs to stabilize the new capillaries. This image illustrates the process of angiogenesis, showing the formation of new blood vessels from existing one, which is essential in tissue healing and vascular growth. This is capturing the stepwise event that we have already discussed. Uh, the stepwise events of the angiogenesis and the dynamic interplay between the endothelial cells, pericyte, basement membrane, and signaling factors like VEGF, angiopytins, and matrix metalloproteinases. First, there is a quiescent vessel. Here, this frame of the quiescent vessel, it is showing a resting blood vessel that is lined by these endothelial cells, and it is encased by thin basement membrane, and for outer surrounding pericytes or supporting cells are also present. These are the supporting pericytes. So under the influence of the angiogenic factors, the stimulus for the angiogenesis begins. These angiogenic factors may be VEGF, vascular endothelial growth factor. So there is detachment of the pericyte as well as the vasodilation. So this vasodilation occur under the influence of nitric oxide and VEGF, and with the detachment of the pericyte that is facilitated by angiopytins, and degradation of this basement membrane is facilitated by matrix metalloproteinases. So a leading tip is formed. A leading tip of the endothelial cells is formed. It is guided by VEGF and notch signaling. It begins to sprout. Then the next step is the elongation of the vascular stalk and the recruitment of the pericyte, that is shown in this frame. So there is elongation of the vascular stalk, that is a tubular extension of the endothelial cells. This is the tubular extension of the endothelial cells through the extracellular matrix. Here you can see the pericytes are also recruited outside to support the vessel, and they also responsible for elongation of the vessel. Finally, a new vessel is formed and it matures with the formation of the basement membrane, stabilization of the junctions of the endothelial cells, and complete recruitment of the pericytes, forming a fully functional blood vessels that are non-leaky and do not give passage, easy passage for the flow of the content, as happens in the immature vessels that allow the leakiness. So in the process of angiogenesis, there is continuous receptor crosstalk. If there is a change in the extracellular matrix that is sensed by surface integrin receptors, and integrin and growth factor signal cascade converge to trigger many of the processes such as cell survival, cell proliferation, differentiation of the cells, and migration of the cells. All of these are uh, there to help regulate the key endothelial cell function. So, cell locomotion by organization of the cytoskeletal changes at the membrane occurs. There is also activation of the endothelial cells that causes the new integrin formation or expression that modulate their migration, cell migration on provisional matrix protein. Two such integrin are beta 1 type integrins that are associated with capillary sprouting, and alpha V beta 3 integrins that are also termed as vitronectin receptors, and they are responsible for survival and special organization of the matrix protein, and they respond properly to the extracellular matrix composition and structure. This histology picture is of granulation tissue. The granulation tissue is showing the numerous blood vessels that are highlighted, and these blood vessels, these blood vessels, they are filled with blood, and they are prominent and abundant in granulation tissue. These are filled with red blood cells. Redness is due to red blood cells, and they are embedded in a pale stained extracellular matrix of collagen that is purple blue in this picture. Loose collagen that is initially of type 3, that then it later is replaced with type 1 of the collagen, and the tissue appears loosely organized with numerous red blood cells that are scattered throughout, because we know that initially these blood vessels are leaky also. So most of these vessels have got thin wall. Over here these are thin walled, and some vessels are packed with RBCs, a lot of RBCs, while some vessels are having few of them. Then extracellular matrix that is stained with this purple blue color, they are indicating collagen rich stroma. This wavy fiber presence with irregular arrangement suggests a highly active proliferative phase of typical of granulation tissue. Dark purple blue nuclei uh, of fibroblasts and other stromal cells are scattered throughout the section but not prominent in this field. Their cells density is moderate, that is consistent with early wound healing. So this is a granulation tissue that is characterized by numerous small newly formed blood vessels reflecting angiogenesis, loose collagen matrix, that is early collagen matrix deposition, scattered inflammatory cells that are not very much prominent over here. You can see a few only inflammatory cells. Red blood cells are seen within the new vessels. Some are having a lot of them and some are having few. And there is hyperemia of this section and bleeding tendency in this immature blood vessels. This is the Mason's trichrome staining of the tissue section, which is commonly used to highlight the collagen and differentiate it from other tissue component. So collagen fibers over here are stained blue. All these blue staining is of collagen fibers, uh, while the muscle fiber, cytoplasm, and keratin in this stain appears red. The nuclei, they appear blackish and or dark purple in this stain. So this image is a dense blue stained collagen fiber rich field indicating mature well formed extracellular matrix. The collagen fiber over here, these are wavy and provide support to the structure because they are densely packed. Scattered scattered red stained areas are blood vessels that are filled with RBCs or various blood cells. So this is mature scar tissue or fibrosis as indicated by this dense blue network of wavy fibers. Now the fibroblasts or inflammatory cells are low in number. So this is a late stage wound healing phase or remodeling phase where the collagen matrix is reorganized and cross-linked to form a strong scar. Remember, in Mason's trichrome, this collagen appears blue color and it is typical of mature scar that have high collagen content and low cellularity compared to the early granulation tissue. Blood vessels are sparse and appear as red stained structures over here. Thank you for watching this video. If you have any question, please write in the comment box.